Hi, I'm Jasper Eaton, European Sales Representative for Andrew Alliance. Today, I want to share with you an exciting new application on our Andrew Plus pipetting robot. Andrew Alliance has been working with its partner, Nagel, to achieve large volume plasmid DNA purification by automating their NucleoBond Xtra Midi prep kit. There is strong demand for plasmid DNA in the production of next-gen cell and gene therapies, as well as vaccines. But it's really tough to automate. So what is the challenge in automating it exactly, and why has nobody done it yet? Well, actually, in order to extract pure plasmid DNA from bacterial pellets, there are 4 challenges. The first is that it requires large buffer volumes, from 8 to 10 ml. And secondly, tall columns are indispensable, and there is complex handling of labware involved as well. Lastly, there's a desalting step critical to the success of the workflow. Consequently, this laborious process has been very difficult to automate up to now. But guess what? We've made it happen. As of today, the NucleoBond Xtra Midi prep workflow can be successfully automated using the award-winning Andrew Plus pipetting robot. I'd like to highlight some key features of the robot. You can see the robotic arm, which can select different pipettes and tools from its tool rack, enabling it to perform a wide range of liquid handling applications in an efficient and highly repeatable way. At the bottom are consumable holders called Dominos, which can be freely combined according to the task at hand. Here's one for the 5 ml pipette tips, one for the 50 ml columns, and one for MidiPrep columns. The robot is set up and controlled remotely by cloud-native OneLab software, which benefits from validated protocols, easily downloaded from the OneLab online library, or set up by the operator using a highly intuitive interface, requiring no knowledge of programming and shared with other members of the lab via the cloud. Let's have a closer look here. At the start of any workflow, the robot scans the layout, confirming the presence of required Dominos, consumables, and any other required accessories. But with all that said, how does the robot solve the 4 challenges of automating the NucleoBond Xtra Midi prep? It starts with the pipettes. The Andrew Plus robot uses high-performance Bluetooth electronic pipettes, enabling a wide range of buffer volumes to be pipetted, up to those 10 ml. Oh, and OneLab programs the settings of the electronic pipettes remotely via Bluetooth for quick volume changes and repeatable, precise pipetting. Tall columns can be moved due to the high overhead transfer capability of Andrew Plus. Our new column domino has space for 3 columns, enabling the transfer of large buffer volumes, capture and removal of waste buffer, and autoclavability. Additional column dominoes can increase sample throughput as well. A new Bluetooth connected tool, called the column gripper, can move MidiPrep columns from the wash position to an elution vessel for plasmid elution. OneLab enables additional ontologies. Tube Magnet Plus automates the required desalting step using magnetic bead separation, giving Andrew Plus incredible flexibility. OneLab and the Andrew Plus pipetting robot have successfully automated the NucleoBond Xtra Midi prep, enabling large volume, repeatable production of plasmid DNA without compromising the yield, purity, and integrity. Additionally, each step of the protocol is recorded by OneLab, ensuring full traceability. Thanks a lot for your attention. You can get more information on our website, andrewalliance.com.